Rapid life cycles of marine phytoplankton transfer heat-trapping carbon dioxide, CO2, from the atmosphere and upper ocean to the deep sea, where the gas remains sequestered until currents return it to the surface hundreds of years later. If all of the world's marine phytoplankton were to die today, the concentration of CO2 in the atmosphere would rise by 200 parts per million or 35% in a matter of centuries. Adding certain nutrients to the ocean surface can dramatically enhance the growth of phytoplankton and thus their uptake of CO2 via photosynthesis, but whether intentional fertilization increases CO2 storage in the deep sea is still uncertain. Artificially enhancing phytoplankton growth will have inevitable but unpredictable consequences on natural marine ecosystems. Every drop of water in the top 100 meters of the ocean contains thousands of free-floating, microscopic auricalled phytoplankton. These single-celled organisms including diatoms and other algae inhabit 3/4 of the Earth's surface, and yet they account for less than 1% of the 600 billion metric tons of carbon contained within its photosynthetic biomass. But being small doesn't stop this virtually invisible forest from making a bold mark on the planet's most critical natural cycles. Arguably one of the most consequential activities of marine phytoplankton is their influence on climate. Until recently, however, few researchers appreciated the degree to which these diminutive ocean dwellers can draw the greenhouse gas carbon dioxide, CO2, out of the atmosphere and store it in the deep sea. New satellite observations and extensive oceanographic research projects are now revealing how sensitive these organisms are to changes in global temperatures, ocean circulation, and nutrient availability. With this knowledge has come a temptation among certain researchers, entrepreneurs, and policymakers to manipulate phytoplankton populations by adding nutrients to the oceans in an effort to mitigate global warming. A 2-month experiment conducted early this year in the Southern Ocean confirmed that injecting surface waters with trace amounts of iron stimulates phytoplankton growth. However, the efficacy and prudence of widespread, commercial ocean fertilization schemes are still hotly debated. Exploring how human activities can alter phytoplankton's impact on the planet's carbon cycle is crucial for predicting the long-term ecological side effects of such actions. Seeing green over time spans of decades to centuries, plants play a major role in pulling CO2 out of the atmosphere. Such has been the case since about 3 billion years ago, when oxygenic, or oxygen-producing, photosynthesis evolved in cyanobacteria, the world's most abundant type of phytoplankton. Phytoplankton and all land-dwelling plants which evolved from phytoplankton about 500 million years ago use the energy in sunlight to split water molecules into atoms of hydrogen and oxygen. The oxygen is liberated as a waste product and makes possible all animal life on Earth, including our own. The planet's cycle of carbon, and, to a large extent, its climate, depends on photosynthetic organisms using the hydrogen to help convert the inorganic carbon in CO2 into organic matter, the sugars, amino acids and other biological molecules that make up their cells. This conversion of CO2 into organic matter, also known as primary production, has not always been easy to measure. Until about 20 years ago, most biologists were greatly underestimating the contribution of phytoplankton relative to that of land-dwelling plants. In the second half of the 20th century, biological oceanographers made thousands of individual measurements of phytoplankton productivity. But these data points were scattered so unevenly around the world that the coverage in any given month or year remained extremely small. Even with the help of mathematical models to fill in the gaps, estimates of total global productivity were unreliable. That changed in 1997, when NASA launched the Sea Wide Field Sensor, CWIFS, the RST satellite capable of observing the entire planet's phytoplankton populations every single week. The ability of satellites to see these organisms exploits the fact that oxygenic photosynthesis works only in the presence of chlorophyll A. This and other pigments absorb the blue and green wavelengths of sunlight, whereas water molecules scatter them. The more phytoplankton soaking up sunlight in a given area, the darker that part of the ocean looks to an observer in space. A simple satellite measurement of the ratio of blue-green light leaving the oceans is thus a way to quantify chlorophyll and, By association, phytoplankton abundance. The satellite images of chlorophyll, coupled with the thousands of productivity measurements, dramatically improved mathematical estimates of overall primary productivity in the oceans. Although various research groups differed in their analytical approaches, by 1998 they had all arrived at the same startling conclusion: every year phytoplankton incorporate approximately 45 billion to 50 billion metric tons of inorganic carbon into their cells, nearly double the amount cited in the most liberal of previous estimates. That same year my colleagues Christopher B. Field and James T. Randerson of the Carnegie Institution of Washington and Michael J. Bierenfeld of Rutgers University and I decided to put this GUR into a worldwide context by constructing the RST satellite-based maps that compared primary production in the oceans with that on land. Earlier investigations had suggested that land plants assimilate as much as 100 billion metric tons of inorganic carbon a year. To the surprise of many ecologists, our satellite analysis revealed that they assimilate only about 52 billion metric tons. In other words, phytoplankton draw nearly as much CO2 out of the atmosphere and oceans through photosynthesis as do trees, grasses, and all other land plants combined. Sinking out of sight, learning that phytoplankton were twice as productive as previously thought meant that biologists had to reconsider dead phytoplankton's ultimate fate, which strongly modifies the planet's cycle of carbon and CO2 gas. Because phytoplankton direct virtually all the energy they harvest from the sun toward photosynthesis and reproduction, the entire marine population can replace itself every week. In contrast, land plants must invest copious energy to build wood, leaves and roots and take an average of 20 years to replace themselves. As phytoplankton cells divide every 6 days on average, half the daughter cells die or are eaten by zooplankton, miniature animals that in turn provide food for shrimp, sh and larger carnivores. The knowledge that the rapid life cycle of phytoplankton is the key to their ability to enhance climate-inspired and ongoing international research program called the Joint Global Ocean Flux Study, JGOFS. Beginning in 1988, JGOFS investigators began quantifying the oceanic carbon cycle, in which the organic matter in the dead phytoplankton cells and animals' fecal material sinks and is consumed by microbes that convert it back into inorganic nutrients, including CO2. Much of this recycling happens in the sunlit layer of the ocean, where the CO2 is instantly available to be photosynthesized or absorbed back into the atmosphere. The entire volume of gases in the atmosphere is exchanged with those dissolved in the upper ocean every 6 years or so. Most essential to climate is the organic matter that sinks into the deep ocean before it decays. When released below about 200 meters, CO2 stays put for much longer because the colder temperature and higher density of this water prevents it from mixing with the warmer waters above. Through this process, known as the biological pump, phytoplankton remove CO2 from the surface waters and atmosphere and store it in the deep ocean. Last year, Edward A. Laws of the University of Hawaii, 3 other JGOFS researchers and I reported that the material pumped into the deep sea amounts to between 7 billion and 8 billion metric tons, or 15%, of the carbon that phytoplankton assimilate every year. Within a few hundred years, almost all the nutrients released in the deep sea and their way via upwelling and other ocean currents back to sunlit surface water, where they stimulate additional phytoplankton growth. This cycle keeps the biological pump at a natural equilibrium in which the concentration of CO2 in the atmosphere is about 200 parts per million lower than it would be otherwise, a significant factor considering that today's CO2 concentration is about 365 parts per million. Over millions of years, however, the biological pump leaks slowly. About 0.5% of the dead phytoplankton cells and fecal matter settles into CaO or sediments before it can be recycled in the upper ocean. Some of this carbon becomes incorporated into sedimentary rocks such as black shales, the largest reservoir of organic matter on Earth. An even smaller fraction forms deposits of petroleum and natural gas. Indeed, these primary fuels of the industrial world are nothing more than the fossilized remains of phytoplankton. The carbon in shales and other rocks returns to the atmosphere as CO2 only after the host rocks plunge deep into the Earth's interior when tectonic plates collide at subduction zones. Their extreme heat and pressure melt the rocks and thus force out some of the CO2 gas, which is eventually released by way of volcanic eruptions. By burning fossil fuels, people are bringing buried carbon back into circulation about 1 million times faster than volcanoes do. Forests and phytoplankton cannot absorb CO2 fast enough to keep pace with these increases, and atmospheric concentrations of this greenhouse gas have risen rapidly, thereby almost certainly contributing significantly to the global warming trend of the past 50 years. As policymakers began looking in the early 1990s for ways to make up for this shortfall, they turned to the oceans, which have the potential to hold all the CO2 emitted by the burning of fossil fuels. Several researchers and private corporations proposed that artificially accelerating the biological pump could take advantage of this extra storage capacity. Hypothetically, this enhancement could be achieved in two ways: add extra nutrients to the upper ocean or ensure that nutrients not fully consumed are used more efficiently. Either way, many speculated, more phytoplankton would grow and more dead cells would be available to carry carbon into the deep ocean. Fixes and limits until major discoveries over the past 10 years cleared the natural distribution of nutrients in the oceans. Scientists knew little about which ocean fertilizers would work for phytoplankton. Of the two primary nutrients that all phytoplankton need, nitrogen and phosphorus, phosphorus was long thought to be the harder to come by. Essential for synthesis of nucleic acids, phosphorus occurs exclusively in phosphate minerals within continental rocks and thus enters the oceans only via freshwater runoff such as rivers. Nitrogen, N2, is the most abundant gas in the Earth's atmosphere and dissolves freely in seawater. By the early 1980s, however, biological oceanographers had begun to realize that they were overestimating the rate at which nitrogen becomes available for use by living organisms. The vast majority of phytoplankton can use nitrogen to build proteins only after it is XED, in other words, combined with hydrogen or oxygen atoms to form ammonium, NH4+, nitrite, NO2, or nitrate, NO3. The vast majority of nitrogen is XED by small sets of bacteria and cyanobacteria that convert N2 to ammonium, which is released into seawater as the organisms die and decay. Within the details of that chemical transformation lie the reasons why phytoplankton growth is almost always limited by the availability of nitrogen. To catalyze the reaction, both bacteria and cyanobacteria use nitrogenase, an enzyme that relies on another element, iron, to transfer electrons. In cyanobacteria, the primary energy source for nitrogenization is another process that requires a large investment of iron, the production of adenosine triphosphate, ATP. For these reasons, many oceanographers think that iron controls how much nitrogen these special organisms can X. In the mid-1980s, the late John Martin, a chemist at the Moss Landing Marine Laboratories in California, hypothesized that the availability of iron is low enough in many ocean realms that phytoplankton production is severely restricted. Using extremely sensitive methods to measure the metal, he discovered that its concentration in the equatorial Pacific, the northeastern Pacific and the Southern Ocean is so low that phosphorus and nitrogen in these surface waters are never used up. Martin's iron hypothesis was initially controversial, in part because previous ocean measurements, which turned out to be contaminated, had suggested that iron was plentiful. But Martin and his co-workers pointed out that practically the only way iron reaches the surface waters of the open ocean is via windblown dust.